Welcome, everyone. Thanks for stopping by 17 Minutes of Science. My name is Sarah Cheeseman, and I'm a technical solutions scientist at In Vivo Biosystems located in Eugene, Oregon. Today, I'm really excited to welcome our guest, Mark Mendoza, to the show. And we're going to talk about a very timely topic, which is viral gene therapies and their potential use against COVID-19. So in this moment where we know it's all hands on deck to identify therapeutics to fight this novel coronavirus, I know our viewers and I am very interested to learn more about how your team and others are exploring a harmless virus mechanism to fight a harmful virus, which is a little different than what I think we're hearing a lot of the time, which is around small molecules. So I'll say a few words about Mark and then I'll let him introduce himself and then we'll jump right into the questions. So Mark is a scientist in Hans-Peter Kiem's lab at the Fred Hutch Cancer Research Center up in Seattle, where they use gene therapy and gene editing of hematopoietic stem cells to treat and cure blood disorders and HIV. And Mark is involved in developing research grade and clinical grade viral vectors for translational applications. So here's Mark. Would you say a few words about yourself?
Yeah, so thank you, Sarah. I'm very excited to be conversing with you. So I've been a researcher within the Kiem lab for about a couple years. And so I've really been enjoying the vast projects that we get to be a part of. And I think that it's very timely, even working here at Fred Hutch, where working everything from cancer research to also having a division studying infectious disease. Well, with the rise in COVID cases, I'm seeing day to day, week to week, the sorts of solutions that our scientists are coming up with. So I'm very fortunate to be in such a great environment.
Well, thanks for joining us today to talk about this topic, which is so interesting and worthy of more information being shared about it. So can we start at the beginning, which is to say, what is a viral gene therapy? Can you help us define that?
Yes, so a viral gene therapy is nothing new. Maybe the word virus these days, a little taboo, but viral gene therapy is basically a vehicle or a delivery method to traditionally you can use a lentiviral vector that will integrate a piece of DNA or a nucleic acid into a host genome. And so that can then repair, let's say, a non-functioning gene. And so the way that these viruses are able to, in a way, hijack the host's machinery, you can think of it as, in this case, it's using it for good. And these therapies are long-lasting. And so the part of what's being developed is the safety, because safety is number one. And so they're also developing what are called adeno-associated vectors or viral vectors, which do not integrate into the host genome. So those are another safety mechanism. And so we're seeing that more and more in the research lab and potentially in the clinic one day.
So is that the direction things are headed right now? So in terms of how viral gene therapy can be used against COVID-19, is it through these adenovirus associated vector approaches?
Yeah, that's correct. So one research team right now, and I know that this is still it amazes me how fast we're moving. Before 2020, back in 2019, we're using the concept of an adeno-associated virus, or AAV, for an array of applications. And so now with the biologist toolbox or toolkit, we're able to harness the power of CRISPR as well. And so essentially you could get what it is that you want to get introduced into the host cell. In this case, if it's an epithelial cell, which is where the coronavirus is able to take a hold from the receptor, the ACE2 receptor in these epithelial cells. And so you could use these viral vectors to then make these copies of what you want. In this case, it would be CRISPR RNA. And so in a way, this CRISPR molecule will then bind to the target RNA and then degrade it. So there's no harm in the sense that it will integrate as a traditional virus would, but it's still in the early stages, and it's promising with what's being done in vitro studies. And so we'll see what comes out of it, if it can hopefully be sped up and go into the clinic and into clinical studies.
Being that those AAVs are attacking the SARS-CoV-2 at that cell type, you're trying to intercept that before they actually bind and enter the host cell and begin to wreak havoc?
Yeah, and even if they already do, so what they're wanting to harness out of this is find a conserved region, because it is to know that this virus and a host of other viruses have mutations, and they tend to change. So if you can find a nice conserved sequence, you could maybe attack many other sorts of coronaviruses. So they're going off of coronaviruses that we've seen in the past, coronavirus that we're seeing now, this novel coronavirus. And so it will find this RNA, and so because the primary nucleic acid that carries all the instructions, which is for these coronaviruses RNA-based, so it relies on producing its own RNA polymerase. And so what the CRISPR technology can do with these AAVs is target the mRNAs or the RNAs that they have. And if they have already gone into producing these mRNAs, they can bind whatever it is, this complementary strand. And just like what we've known about CRISPR, is it can cleave it, degrade, and hopefully that will be the end of the story with what the coronavirus is then on track to do.
That's so fascinating. In terms of gene therapy broadly, in this case in particular, but are there side effects for patients for this kind of approach?
Yes. So much of gene therapy right now at the moment is done not in vivo, but ex vivo. So a lot of the work here at Fred Hutch, the pioneering work in the beginning, was with bone marrow transplantations. And so with the type of work that we do in our lab, what we typically do is we get we remove stem cells from the host, from the patient, and then we introduce a viral vector that is carrying what it is that we want to insert into the host genome. And so going along that route, we have to make sure that there is a HLA matching because we don't want any graft versus host rejection or disease to come about that. So we could also do, so we can find someone who is a match, or we could also remove stem cells from themselves. And so sometimes with some of these therapies, especially with gene therapy, is we have to make room in the bone marrow. And so they'll do what's called conditioning, oftentimes referred to as chemotherapy, or different chemotherapy agents, to make room in the bone marrow, so that we can, once we've removed those stem cells, treated it and have them expand, and then we can then put them back into the patient, and those cells will be the more functional type in order to reconstitute that space.
So are those the kinds of things then, of course, you're considering as the field is trying to tackle COVID-19 through this therapy? Have to take all that.
Yeah, I'll have to, I guess, do a bit more research as to what the strategy is going to be with COVID-19 and how they're going to approach that. So of course they wouldn't be looking at bone marrow cells as they would be epithelial cells. And so ideally, the goal would be an in vivo therapy. So this would be more of an antiviral therapy in which an injection could potentially just eliminate the RNA that transmits the disease. And because the viruses, they have one mission, and it is to replicate as much as possible.
Right. It's just to try and shut that train down.
Yes.
It's, on the tracks, so to speak. Gosh, so many questions. Let's see. Trying to think of in terms of Fred Hutch itself as an institution. I mean, you were just speaking to how its main focus is on cancer research and treatment. And so how are you seeing the intersection of the work that you're doing on a day-to-day with this emergence of this novel virus and members of your research team being involved in that? What's that like in the halls right now?
Yeah, so I will say that I don't have it on screen, but everyone, I haven't really seen many people's faces. We're all fully covered with our face masks. So it's like a whole new identity. But we've been taking it very seriously, from having the right protection and also the proper precautions. We're doing testing here on campus. I've already been tested and screened a couple of times. So as we are working as an institute to help develop a vaccine and to even track the virus as it happens, not only here in Seattle, but also globally, we are also looking, as we're getting tested, into developing a study, or actually it's already in the works. So I'm already a part of that study. And so we're looking at our employees, and I will say that from the first study that was produced, we didn't have anybody that tested positive. There were some antibody cases, people who tested positive for antibodies, meaning that the employees, I would expect this from scientists, to be very cautious. But now as the world is starting to open up, we'll see if we can hold that consistency. We have several researchers, Dr. Larry Corey, one of them, he's working with Dr. Anthony Fauci. And so they are collaborating on potentially bringing a vaccine, hopefully by December 2021. So it still seems like it's pretty far away, but there are also potential therapies, antiviral and vaccinations still in development. And I think that what's really important is for people to remember that as the world does open back up, that the virus is still around, and to be as cautious as possible. At least that's what they're having us do here at the Institute or at the center. In addition, I think it's great that we also have a great bioinformatics team that is tracking what is going on, the number of cases, if there are different strains, where they're originating. And so the genomics core and the sequencing, I think, is a huge tool and a big player in helping us understand the virus as it's spreading.
Well, it certainly is terrific that you have all those resources there to leverage against this whole new research direction, in addition to your core competencies. I mean, that's exactly what we need right now, is for that kind of agility among our scientific community. We had a great question come in from the co-founder of In Vivo Biosystems. So I'd like to share it with you, which is, how is the regulatory environment for gene therapies? Is it too strict? Is it too relaxed? Is it just right? Can you comment on that?
I think that from what I've seen, it's at the moment, it's just right. I think that stringency with regulatory is necessary for what has happened in the past, in the early days of viral therapies. So part of the tricky part about these viral vectors is that they can integrate anywhere, at least with a lentiviral vector. With the vectors that have already been in clinical trials and potentially on the market are the adenoviruses, so separate from the adeno-associated viruses, the AAVs I mentioned earlier. So these adenoviruses can predictably integrate into chromosome 19, but a lentiviral vector can integrate itself into anywhere. So because of that, we have to be very cautious as to where they integrate and that they don't integrate into potentially a proto-oncogene that can then lead to cancer as there is expansion of these cells. And so it could be more of a problem than a solution to what the patient is going through. So some of what has to happen, some of what I even do as well, is doing a series of tests, everything from sequencing to the quality of these viral vectors, stability tests years on out, if it's going to be stored away, take it out of the freezer 1 year later, 3 years later, making sure that the efficacy of the vector is something that is safe for the public. And that is a major consideration that we look at in our preclinical models.
Fascinating. Well, that's a great question, and thanks for the audience member.
Absolutely.
I forgot to mention we have a timer going. I didn't give you a heads up because we just did it. That was our 17 minutes.
Oh my gosh, it flew by.
I was so excited to start a conversation. I haven't been good lately about giving people the heads up that I'm pressing. It sneaks up on us at the end. But Mark, I really want to thank you for taking the time today to talk to us about this fascinating angle as we think about what we need in our therapeutic toolbox to move forward as a world in a COVID-19 world that we're going to be living in for a while. So I wish you all the best. It's fascinating, and I hope that you'll stay in touch with us and let us know how it's going.
Absolutely, yes. Everyone take care of themselves and each other, and I'm optimistic as to what can come out of the science field. Everything from basic research to what we're doing in translational science as well.
Absolutely. Well, that's the perfect end note. So we'll leave it there. Thank you again. Thanks everybody for tuning in, and we will see you next week.
Thank you.